Second kidney transplant from a genetically modified pig. New York surgeons performed the second ever operation to transplant a kidney from a genetically modified pig into a living human. This time, the procedure was performed in conjunction with the implantation of a device that assists the functioning of the heart ventricles. The patient, a 54-year-old woman with heart and kidney failure, is feeling well after the surgery and is recovering. In late March, surgeons at Massachusetts General Hospital announced that they had performed the world's first kidney transplant from a genetically modified pig to a 62-year-old man with end-stage kidney disease. The patient, Richard Slayman, was discharged home after a few days and, according to his doctors, he is now feeling well. The second person to receive a kidney from a genetically modified pig was 54-year-old Lisa Pisano. The woman struggled with heart failure and end-stage kidney disease, which required routine dialysis. Her condition was getting worse and she was not eligible for a heart or kidney transplant. The pool of organs available for transplantation is limited and there are many more people willing to do so. So those who are eligible for transplantation are those who have the greatest chance of transplant success. The woman actually had no treatment options left. But doctors from New York University Langone Health managed to develop a pioneering, two-part surgery that involved implanting an electromechanical pump, i.e. a ventricular assist device, VAD. And after a few days after transplanting a kidney from a genetically modified pig, along with a thymus with genes modified to prevent rejection. After I was excluded from the possibility of a human organ transplant, I learned that I didn't have much time left. But my doctors informed me that there was a chance of receiving a kidney from a pig from a genetically modified herd, so I discussed it with my family and husband, Pisano said. A statement on the NYU Langone Health website states that so far there have been no documented cases of transplants to people with VAD. This is also only the second transplant of a pig kidney with modified genes into a living person and the first including the thymus. Before scientists decided to transplant the first pig kidney into a human, they tested the concept on a patient with a brain dead and symptoms of kidney dysfunction, whose family consented to the experiment before she was to be disconnected from life support. Surgeons attached a pig kidney to the patient's blood vessels and kept it outside her body for 3 days. This gave scientists the opportunity to have unlimited access to the organ and thoroughly examine the kidney. It's incredible when you consider the science that allowed us to save Lisa's life and what we strive to do as a society for everyone in need of a life-saving organ, said Robert Montgomery, who led the surgical teams. This could not have been achieved without the dedication and skill of the many talented physicians, researchers, nurses, healthcare administrators and perioperative care teams at NYU Langone Health, as well as the numerous pioneers who have come before us, he added. The series of procedures were performed by 2 separate teams over 9 days. During the first procedure, surgeons implanted a heart pump that assists the left ventricle, or LVAD. This device is typically used in patients who are waiting for a heart transplant, or are otherwise ineligible for a heart transplant. Doctors acknowledged that without the LVAD, Pisano's life expectancy would have been measured in days or weeks. The second operation was xenotransplantation. This term refers to the transplantation of organs or tissues from an individual belonging to another species. Along with the kidney, the patient also received a thymus transplant from a pig. The thymus gland is essential for the proper development of the body's immunity. It was transplanted along with the kidney to educate the 54-year-old's immune system. The very concept of a thymus kidney transplant was intended to reduce the amount of immunosuppressive drugs needed after transplantation by teaching the immune system not to attack the new kidney. Pisano's surgery was successful. According to doctors, the transplanted kidney began producing urine almost immediately. Pisano also started walking, which was almost impossible for her before the surgery. However, Montgomery says the critical moment comes about a month after surgery, so the patient will be closely monitored. The herd of genetically modified pigs was developed by Revivicor, a subsidiary of United Therapeutics. In total, about 100 modified animals were created. They live in strictly controlled conditions on a farm in Iowa, USA. They do not have the gene that produces alpha-gal, a sugar that triggers an immediate attack by the human immune system. But this is not the only change in the pig genome. Using the CRISPR-Cas9 gene editing method, a total of 69 changes were introduced into pigs. Some pig genes have been deactivated, others modified. In addition, several human genes were introduced into the pig genome. These modifications were intended to make pig organs more tolerant to the human immune system. Xenotransplantations may address the shortage of human organs in the future. Contrary to appearances, human genetic material does not differ significantly from pig genetic material. The size of the organs and the mechanism of action are also appropriate. There is a huge demand for organs for transplantation, but there are too few organs available. In recent years, scientists have made great progress in transplanting organs from pigs to humans. First, the organs were transplanted into primates. When these trials showed promising results, they began experimenting with humans. The most famous cases were 2 heart transplants from pigs. The first patient, David Bennett, died 2 months after the transplant. He did well at first. He began physical therapy to regain his strength, but after about 6 weeks his condition worsened and he died a few days later. The hospital said in a statement that the patient died due to multiple factors and general poor health. It later turned out that the transplanted heart contained a pig-infecting virus that may have played a large role in the patient's death. The second patient who received a pig heart, Lawrence Fawcett, died 6 weeks after surgery. It is currently unknown exactly what caused the death. Doctors from the University of Maryland Medical Center wrote in a statement that the patient was making significant progress after surgery and was undergoing physical therapy. But a few days before his death, the first signs of rejection of the pig heart began to appear. The medical team did everything they could, but they could not save the man's life. It should be noted that both men suffered from very serious heart diseases and, according to doctors, they faced almost certain death due to heart failure. bees from Africa that can create clones of themselves. They may have millions of copies. A group of scientists have discovered that certain honeybees native to South Africa can create identical copies of themselves. Moreover, one individual can clone itself millions of times, an ability that allows it to take over the hives of its rivals. An invisible, internal enemy is responsible for the destruction of the hives of the African honeybee, Apis mellifera scutella. It's about a growing, seemingly indestructible army of clones of a competing subgenre. All because the workers of a competing subspecies, the South African honeybee, Apis mellifera capensis, also known as the Cape, can create perfect copies of themselves. One individual has cloned itself millions of times over the past 3 decades. According to a study published in the journal Proceedings of the Royal Society Bee with the ability to create clones of themselves, South African bees sneak into the hives of their lowland rivals and produce copies of themselves inside, taking over the hive. Worse still, these clones are often freeloaders and refuse to do any work. Asexual reproduction, parthenogenesis, is not uncommon in the insect world, but having offspring genetically identical to the parent is. This happens because during reproduction, genetic material mixes in a process called recombination. As a result, even if there is only one parent, its offspring have a slightly different genetic makeup. However, Cape bees in South Africa have developed the extraordinary ability to clone themselves while successfully avoiding recombination during reproduction. This allows them to consistently recreate a perfect copy of themselves, or simply create clones. According to researchers, bypassing the process of reshuffling the gene pool is unheard of. It's amazing, but also incredibly dysfunctional, says the lead author of the discovery, Professor Benjamin Oldroyd from the University of Sydney. And yet, somehow the bees manage to lay eggs without reshuffling the gene pool. Stem cell. This is crazy. I haven't heard of anything like this before, he adds. As already mentioned, honeybee workers and other social insects have the ability to reproduce asexually. Females produce female offspring from unfertilized eggs. This rare type of parthenogenesis, called thelitoky, has only been observed in about 1,500 species. However, each time this process is accompanied by a reshuffling of the gene pool. This ensures that even with one parent, future offspring will be genetically diverse. However, nothing can replace sexual reproduction. Each new generation of workers reproducing in this way loses about 1/3 of their genetic diversity, which can lead to death after a few generations. Therefore, most social insects rely on a queen to reproduce sexually, as it were, on their behalf. In return, genetically diverse workers keep the colony healthy and protect the offspring. Just like in human society, we have a tension between what is good for the individual and what is good for society. And we come up with social norms that allow us to function, Oldroyd says. One of the things in honeybee societies that has evolved to suppress selfish behavior is that worker bees generally cannot lay eggs, he adds. According to the expert, similar rules also apply to the Cape bee, which generally reproduces like other social insects. However, the workers of this subspecies have a mutation that allows them to lay eggs with all their genetic material, without any reshuffling. With this ability, they can prevent generational loss of genetic diversity. In other words, mutation allows insects to clone themselves whenever they want for decades. Even if in the long run it results in a population that has no overall genetic diversity. This superpower of cloning disturbs the balance between individualism and community, which may ultimately lead to the extinction of the species, say researchers. To understand how clones can make millions of copies of themselves and still remain functional, Oldroyd and his team compared the genomes of Cape worker bees with those of the queen and her offspring. In total, the team examined the DNA of several dozen larvae hatched from eggs laid by the queen and workers of the South African bee. The larvae hatched through asexual reproduction. The researchers found that the level of recombination in the queen's offspring was 100 times higher than in the genetically identical cloned offspring of the workers. This suggests that the worker bees have evolved a mutation that prevents DNA recombination, which in turn allows them to continually make copies of themselves. The ability of workers to clone at will puts their colonies in a precarious situation, especially if the queen leaves or dies. Instead of devoting their energy to getting the colony back on its feet, the workers have their own selfish agendas, such as finding a way to place their clones in positions of power. If, for example, we take the queen out of the hive, instead of raising a new queen, as other species of bees do, Cape worker bees will start laying eggs themselves, says Oldroyd. The researcher also believes that workers of this species can throw eggs into cells containing the larvae of future queens. One line of Cape worker bees took social parasitism even further. The insects no longer need a queen and exist solely by taking over the hives of the African honeybee. The clones sneak into the hives of competing bees and lay as many eggs as they can. The owners of the hive can fuse the eggs with their own and take care of them. The larvae then send signals to feed them as much as possible, which causes them to enlarge their bodies and grow to almost the size of the queen. Cape bee clones do no work inside the hive because they have become reproductive, Oldroyd says. They just walk around the hive and feed on the work of others. This very quickly leads to the collapse of the hive. As individuals, these clones are quite dysfunctional. So you would expect them to destroy themselves. But in this respect they resemble cancer cells, adds the scientist. According to Oldroyd, the Cape worker bees that engage in this parasitism are genetically identical descendants of a single worker who lived in 1990. This single line of clones is responsible for the collapse of 10% of African honeybee colonies each year. Scientists want to conduct further research into hive collapse and worker cloning in Africa. They hope to find mechanisms that enable or disable the ability to clone insects. A system has been discovered in which planets orbit two suns. Astronomers announced the discovery of the second planetary system in history, in the center of which, instead of one star, as in our solar system, there are two suns, and at least two planets revolve around them. Similar objects are called Tatooine, named after the planet from Star Wars that orbits a binary system. At the center of the Toy 1338 system, also known as Bebop 1, is a double star. It is actually a system of two stars orbiting around a common center of mass. A group of scientists led by scientists from the University of Birmingham identified two planets in the system. These are the so-called planets circumbinary, i.e., orbiting both components of the double star. From their surface you could watch two sunrises and two sunsets. Just like on the homeworld of the Skywalker family from Star Wars, the planet Tatooine. The description and results of the research were published in the journal, Nature Astronomy. Binary systems are by no means rare in space. On the contrary, there are actually a lot of them. However, understanding how planets find their way in such systems is still a challenge for us and requires acquiring much more knowledge. A great opportunity in this context is the TOI 1338 or Bebop 1 system. In general, planets can also exist in binary star systems. Typically, however, the worlds orbit one of the components of a binary star, and the gravitational field of the second component, usually far away from the first, has only a small effect on the planets. And in this respect the TOI 1338, Bebop 1 system is completely different. Not only do planets exist in it, but they orbit both stars simultaneously. Up to a certain point, it was even doubted whether something like this could exist at all. Because the evolution of such a system would inevitably lead to the forces acting in such a system leading to the ejection of such a planet into interstellar space. So far, only 12 similar binary systems have been discovered, and only one of them contains more than one planet. There is an interesting story associated with the discovery of two planets orbiting in this system. First of all, this was possible thanks to the TESS Space Telescope. It was with its help that the first planet, designated TOI 1338b, was discovered. It was noticed by a student who was only on his third day of an internship at NASA. However, the capabilities of TESS are limited. Scientists used the observatory to determine the presence of the planet and determine its orbit, but unfortunately it was not possible to determine its mass. Therefore, it was decided to fill this gap. The method of measuring radial velocities was used. However, in this case, scientists also failed because they failed to determine the mass of the discovered planet. The interaction of two stars complicates the situation too much. However, the researchers' work was not in vain, because during the measurements they found another planet in this system, called BEBOP1c. In her case, the weight determination was successful. This planet is approximately twice as far from its suns as TOI-1338b. It takes it 215 days to circumnavigate them. She is also much larger than her companion. Its mass is 65 times that of Earth, or about 5 times that of Jupiter. Both planets in the system are gas giants, but they are also too close to the stars to support life. Interestingly, astronomers believe that there may be even more planets in this system. However, they may be difficult to discover unless they prove to be as massive as Bebop 1c. Perhaps, however, astronomers will be lucky in this matter as well. The system itself is approximately 1,320 light-years away from Earth. It is located in the constellation of the Painter. But this constellation is not visible from our country. At its center there is a binary star. consisting of a yellow-white dwarf slightly larger than our Sun and a red dwarf with a mass about one-third of the Sun. The stars orbit each other every 14.6 days.